Okay, so let's get started. Welcome back to the afternoon session of the Bio-Ontology track. We will, in this session, have a number of short talks and flash presentations. We will have 2 longer talks, and we have our second keynote for the day at the end of the session. So we will start with Jason Hilton, who will talk about ontology applications of the ENCODE portal.
Thank you very much. I am happy to be here representing the ENCODE Data Coordination Center. And if I can just get this full screen.
Control L.
Control L. If I can get it full screen on there. Any thoughts?
I have not. That is out of my area of expertise. Can you help me?
Should we just turn the screen around and I can Now Control L, or do that.
No.
Or Control L. Okay. All right, thank you. So this is a short talk, now shorter. So I want to make sure I get in the acknowledgement of the rest of the ENCODE DCC. I especially want to point out Venkat down there at the bottom, a former member who laid a lot of the framework for what ontologies we use, and he published that in database a couple years ago. Excuse me, a couple years ago. Just to start, this is one way of accessing ENCODE data at the ENCODE portal. This is our matrix view. Along the y-axis, we have different biosamples. The x-axis are the different assays we have, and then around it, you have different ways of filtering down the data. And if you can, there's 14,000 results here, so we have quite a bit of data. We're about halfway through ENCODE 4, so most of that data has yet to be submitted. So that is an increasing number there. And then across the axis, the x-axis here with the assays, the very last one says and 25 more. So if you click that, you expand it. So we have a lot of different data. The other thing I want to point out here down at the bottom, you can filter by project. So it's not just ENCODE data, and I'll talk for a minute about that. So we have ENCODE production labs that are, each lab is submitting data to us. But we have these other projects that some are actively submitting pre-publication data, just like the ENCODE labs are. But we also have some, notably the Roadmap project, that we went to their archives after the project was done and imported their metadata and data. So we have data coming at us from a lot of different ways. So it's really important. Ontologies really help us to standardize the data so that it is easily accessible and it's understandable to for the scientific community, whether they get it directly from us or if they go through one of the genomic resources that we push the metadata and data to. So now just a quick mention of what ontologies we make use of. This is a very simple metadata schematic that we have. We have an experiment. Each experiment has one or more libraries. Each library has a biosample, and that biosample may or may not have a treatment. So first of all, for experiments, we use the OBI ontology for the assay names. One thing we do is we have a set of about 10 higher level OBI terms that we consider to be assay categories. And so for each of these assay term names, here we have for whole genome by sulfite, we go up the ontology relationships that you can see here in red. And if you hit one of these assay categories, it becomes this assay SLIM. So here we have DNA methylation is this parent term above whole genome by sulfite. And putting that into use, going back to our matrix, they're circled at the top. We've selected transcription. So now our assay axis is limited to just those children terms underneath transcription. So we're getting RNA-seq and other transcription assays. I'm going to skip to biosample. and we do a similar thing with that. So first of all, we have cardiac muscle cell. That's a primary cell. And for primary cells, we use CL, or the cell ontology. Tissues, we use Uberon. For cell lines, we use EFO. And this is the organ SLIM is a similar design that we have for the assay SLIM. We define the higher level Uberon terms, and for all of our biosample term IDs, we traverse the ontology to see which organs this fits under. Going back to our transcription results, if we select lung, you'll see here on the y-axis we have cell lines that are derived from lung tissue or lung cells. We have, in the tissue bar there, we have lung tissue, but we also have the subtissues of lung, and then finally primary cells that are part of lung. Another thing we do is that we look up in the ontology the synonyms of that term. So this is not user input. They submit the term name and term ID, and we do a lookup in the ontology for the synonyms. So this way, if someone, for instance, comes to our portal and they search for cardiomyocyte, they're not going to be left in the dark just because we used the term name instead of the synonym. They will still get these results. And real briefly, we use sequence ontology for library and KEBI for chemical treatments. I won't read through all the summary. If you forgot anything, just come see me at my poster tomorrow. A few future directions I think we are really underutilizing. We have a lot, very rich metadata, and I think we could be making use of some more ontologies that are out there. We are developing a cell type SLIM, very similar to the organ type that we have, where it would be a higher level CL term, like a fibroblast, and it would return all the fibroblasts. Assay tree faceting, we're having some issues now, a very complicated problem with different flavors of RNA and how much do you have to tweak the protocol before you get a new assay term name, and then how do we display that on the portal so that we don't confuse our user, and potentially creating objects for each ontology term ID. So that's more of a schema thing. So thank you very much.
And we would have time for one question. Somebody? Okay. So if there are no